All right, good evening, everybody. And good evening to everybody online. I think we have a great crowd tonight, and we're very much looking forward to this next presentation of the Wayne Theater's signature speaker series. And so I'm Joe Kuiper. I'm director of the Virginia Museum of Natural History, working with the Wayne Theater, working with the Center for Cold Waters Restoration, the South River Science Team. We've been doing this for what, 4 years now, something like that? 6 years? I can't believe it. It's like ever since COVID, everything's kind of run together on us. But this has been a great opportunity for us to learn more about the Shenandoah Valley and the Blue Ridge, which of course is what's informing the museum's effort to put a branch campus right here in downtown Waynesboro. So what you're looking at right now is something that's going to go to the Architectural Review Board on Thursday. And so what does that mean? That means that they are looking at our designs that we've done with Glave Homes, an architecture firm out of Richmond, in terms of developing the next phase of our designs for our branch campus at the corner of Arch and Main, just a short walk from here. So what do you see here in this particular slide? I want you particularly to focus on the picture up there, not quite in the corner, but that one showing kind of the Blue Ridge. You have the green in the foreground, the blue in the background. You see how the Blue Ridge and so forth has gotten its kind of characteristic name. Well, our architects use that as inspiration. So let me position myself just right here, right about here. I'm standing in the South River right now at Constitution Park, now renamed the South River Preserve. So as I'm standing here, kind of elevated a bit, over the South River, you're looking uphill towards the downtown. And this here, again, is at the corner of Arch and Main. Now I'm standing on Main Street right here, if that helps any. So what you see is the front of the museum, and notice they're using some architectural nuances to kind of recreate that characteristic view looking out over the Virginia mountains into the Blue Ridge. So this has been a very exciting time. I will say just briefly, since we're kind of in this preliminary sense, there's more to it than this. We've got other drawings. We also have kind of a design of what the inside layout looks like. And we also have we're getting more and more into the details, and we'll be putting those details out as appropriate, but we have more and more details on what the exhibits will look like. And that's been the great thing about the Signature Speaker Series, is that we are inspired by the stories that we get from this region. So work done by regional scientists from our university partners, by our own curators down in Martinsville and whatnot. The more we learn about Virginia, the more stories we have to tell in the exhibits. So here on Thursday, we're going to get some input from the Architectural Review Board. That then goes to the Department of Engineering and Building, and we begin to refine our design, get our cost estimates, and so forth. So where do we stand? Everyone likes to talk about timeline, right? It's always tough. You don't want to be too confident, but you don't want to be pessimistic. Try to find that Goldilocks zone of kind of explaining where we're at in terms of our design. Well, we have a General Assembly session going on right now. Don't forget, though, we're in the second year of a biennial budget. And that's a tough time to get a project pushed forward as far as capital projects go. But we are working on that. We'll see what happens. That said, the state has also released what's called a capital outlay budget. These are the projects that the state is working on that it will fund, and it has already committed to funding this building. The great thing is that they break this list down into three priority categories, 1 through 3, and we are in category 1. So that means we are in the priority position to get funding to be one of the next upcoming capital projects by the state. So we hope within the next year or so, we're going to have that funding that will allow us to start moving dirt at the bottom of the hill and start erecting a building. And so that's where we're at with this. So for the time being, I'm going to leave it at that. I want to introduce our former chairman of the board of the Virginia Museum of Natural History, Dr. Tom Benzing. He's professor at James Madison University and also is a board member of the Center for Cold Waters Restoration right here in Waynesboro. But that said, I'm going to stick around if you want to ask any specific questions about the building, our procedure and process and that sort of thing. I'd be glad to answer them at that point. But go ahead and give a warm welcome to Dr. Tom Benzing.
Thank you, Joe. And as Joe said, this is a really exciting time. There's a lot of information to share about where we are in the process. I want to encourage you, if you're interested to learn more, to come to the Waynesboro Public Library. I've been hosting third Fridays of every month, an hour, 4 to 5 p.m., where you can just informally come in and we can have a chat about the plans and moving forward. So with that, I also want to recognize that the South River Science Team has migrated to what they're now calling the South River Watershed Coalition. And they are a partner, as is the Wayne Theater and the museum and the Coldwater Center, in supporting these programs. And in particular, they provide us with dinner and for our out-of-town speakers an evening in a local hotel. So now to tonight's talk. From trout streams to the tropics, American eels in mountain streams. And for those that attended the November talk, we talked about landslides. So it seems like we have a theme here about slipping and sliding. And we're going to continue that. Coming up in March, we're going to talk about algae. So I guess it's all about slipping and sliding, and slippery eels is probably something that Andy's heard a lot about over his career. Tonight's speaker is Andy Dollif. Andy is a research fishery biologist and the project leader for Watershed Science and the Center for Aquatic Technology Transfer with the U.S. Forest Service. Please join me in welcoming Andy Dollif.
Can you hear me now? Everything's good? Okay. Well, I'd like to thank wow, it is good. Thank the committee for inviting me tonight. They wined me and dined me. Well, they dined me. I didn't get the wine yet. I guess that's something I have to wait a little bit longer for. And I've been looking forward to this for a little while because this is my first official act since I retired a little over 2 weeks ago from everything that Tom just said about me. So I'm no longer the project leader or any of that stuff. I'm still faculty at Virginia Tech because I haven't told them yet that I've officially retired. It's best that you don't bring all this stuff up too much at one time. And I really am excited to talk today and be able to wave my arms as a retired person without undue allegiance to the USDA Forest Service. That said, I really do appreciate the career that I've had with the Forest Service. They've allowed me to do things that I never thought possible. A little bit about me, I grew up in Maine, a little town called Boothbay Harbor on the coast. Went to University of Maine up to Orono, then went to NC State in zoology, went to Montana State in fish and wildlife conservation. Just as an aside, it's a wonder anybody ever graduates from Montana State. If you like to trout fish and hunt and hike, wow. I mean, the temptations are certainly there. And then I did a postdoc and several years up in Alaska. And because of the resources here in the Southeast, I stayed in Virginia. My original plan was, okay, I got a job. It's in Southwest Virginia. Yeah, that's cool, but this is Alaska. Going to be there 5 years and then back to Alaska. 38 years later, here I am. A lot of stuff happened along the way, not the least of which is the opportunity to work with so many of the people, both in the government, at universities, and private citizens and NGOs. It's just been an amazing career. And this is not a capstone by any means, but I am going to talk a little bit about some of the things that happened along the way that steered me towards the direction of what I'm going to talk the bulk of my time about tonight, that is American eels. And I should thank the Forest Service, the Southern Research Station, which is part of the research arm of the Forest Service, the Center for Aquatic Technology Transfer. This is a group that's dedicated to bringing science to the management realm, taking the science that's been developed, particularly in the aquatic realm, and putting it to work on the ground. The National Park Service, because they've enabled me to work on places like Shenandoah, the Great Smokies, a number of other units of the Park Service. Virginia Tech, of course, where I've enjoyed faculty status for 30 plus years. University of Virginia, a long-time partner on several studies ongoing in Shenandoah Park. George Mason University, work with graduate students on American eels. And NC State University just recently finished up working with a graduate student in November, in fact, from NC State. And that's some of the Puerto Rico work that I'll talk a little bit about in a moment. Okay, so. Oops. Nothing is advancing. Oh, there it goes. Wow, that was a moment of panic. Well, one of the ideal things about working with the Forest Service in research is for a fish person, they don't quite know what to do with us. I mean, we're not trees, you know. It's a little bit out of the wheelhouse of the Forest Service writ large. When I started back in the mid-1980s, there were maybe a dozen fisheries research biologists nationwide. That's Alaska to Virginia. I was one of the first two east of the Mississippi River. So you can imagine what that was like. There just weren't that many people around. And I had really developed a passion for long-term data and how important long-term data can be to answering so many of the questions that vex us today, things like climate change. Back then, back in the early days, 1980s, 1970s, we didn't know so much about climate change. We didn't know about things like acid rain, but we didn't realize that that was really part of that whole phenomenon of climate change. And while acid rain fortunes have waxed and waned, climate change has come to the fore. Long-term data is invaluable for short and long-term forecasting. Don't have to convince, I would guess, most people in this audience of that. The value and need is not consistently appreciated. It's often a victim of budgetary cycles. You've been doing this for 3 years. That's long enough. Cut that out. Do something different. Bootleg budget or opportunistic support preserves some elements but limits scope. And I'll talk a little bit about that. So in other words, you get support for something, you get it going, and then somebody says, that's enough. But if you're sneaky, if you're creative, then you're able to continue a lot of those elements. And I've had a career of being opportunistic, let's say. There's an agreement for need on the goals and consistent support. And by goals and consistent support, I rely on a little help from my friends, folks like the Virginia Department of Game and Inland Fisheries. I forget exactly what it's called now, but like that, or the North Carolina Wildlife Resources Commission, Fish and Wildlife Service, a lot of the other folks that come in and say, yeah, we really need this. This is the kind of stuff that's going to pay some dividends. So I've been able to, come on. Why is this thing? Oh, okay. So here are some examples of the long-term research that the Forest Service has chosen to invest in. Right up front is the Forest Inventory and Analysis. This is a nationwide effort to get a complete inventory of all of the trees, the growing stock, if you will. It's a commodity-focused thing. But that said, there are 130 attributes that are collected along with the species and volume of timber. It's where it is, the species that are growing in the understory, exotic invasives. It's a pretty ambitious program, and it is what we call the 800-pound gorilla in Forest Service research. The states and everybody else really wants to support the FIA, and there's no question of the value of the FIA all the way up to the congressional level. Wildlife, well, threatened and endangered species because they get people's attention. That's something that there's been longer-term research support for. Things like red-cockaded woodpeckers, bats, freshwater mussels, migratory birds, and more recently, some of the freshwater fish species. And then at the physical end of things, things like stream water temperature and chemistry, soils, air, vegetative phenology, which is really important now that we're tracking the effects of climate change, and we're seeing southern species creep north, that kind of thing. The Forest Service has several experimental forests nationwide. I was just recently the project leader over the Coweta Hydrologic Lab, where some of the groundbreaking work on things like best management practices, how to build roads without causing undue sedimentation effects, that sort of thing, have been done. The Santee Experimental Forest, which is down on the coast of South Carolina. Again, I was also the administrator over that forest until 2 weeks ago. So there's a lot of long-term research that isn't so much of fishy nature, but there also is some stuff that went completely blank. I doing wrong? There's only 3 buttons. That one works. Help? I'm sorry? Oh. Yeah. Yes, the one, yeah. So just. Exactly, okay, sorry, okay. So long-term research for me, that's why I said personal, I've really had an interest in the ecology and management of large wood. 29 plus year in the Southern Appalachians, 42 years in Southeast Alaska. Even though I've moved, I've been able to get back periodically and follow on to some of the work that I did my dissertation on in Southeast Alaska. Yeah, it's forestry related, but it's also looking at how the juvenile fish in the streams where some of these original clear cuts were done in Southeast Alaska have fared over the last 50 years. Aquatic organism passage in watersheds of the Eastern National Forests. We've been able to do a lot of work with replacing culverts and different stream crossings, bridges versus fords, all that sort of stuff, and the effect on aquatic critters. Stream water and air temperature monitoring network. We ran until recently a network of 250 streams where we were recording every 20 minutes water temperature over the course of about 8 years. This is what's going to be informing some of the modeling effects as we go forward. These were all putative trout streams, by the way. They were screened for that. They just weren't any stream in particular. They were higher elevation. They were definitely known as trout streams. And brook trout in Virginia, some of the long-term brook trout research in Virginia, Payne Run and Stanton River. Payne Run is less than half an hour from here where we sit. We have a lot of information on brook trout in Payne Run and Stanton River. That's part of a long-term cooperative study with the University of Virginia, Virginia Department of Game and Fish. And American eels in Virginia, 22 plus years. And I'm going to take a little deeper dive into that. Okay. Oh, there it goes. If you were to encapsulate my interests over the last 40 plus years, it's basically in history, habitat, chemistry, catastrophe, and more recently, climate, which I call the ingredients in the recipe for fish production in the Appalachians. Now, I say fish production because it's not all about trout, it's not all about eels, although you might think differently after tonight. It's about these kinds of assemblages. This is a chub, and this is a chub mound. You get as many as 27 species on a typical chub mound. These are found in streams all over Virginia. Very, very critical species. We talk about keystone species. We think about wolves and beavers and all that. Well, the chub, that's a keystone species. If he don't build it, they don't come. Literally, the chub has to build a mound so that the other species find a place to spawn. Really interesting stuff. History-wise, I worked with debris in Alaska. You want to see a lot of debris in streams, go to Alaska. Well, there are places in the southeast. The biggest debris jam I've ever seen is on the Chattooga River in southeast Georgia, northeast Georgia. Here the guy is standing on that, and that's a natural jam as far as we know. This is from a landslide that occurred in Stanton River. I hear you had a talk on this thing a little while ago. We've had a fish study going for the several years prior to that debris flow and for the 20 years since that, 18 years since that debris flow. Bottom line for fish is if you're in the way when the debris flow comes down, you're toast. But over time, things actually go back to normal, and for a while they're even better than they were prior to the debris flow. We won't have a chance to talk about that, but if you want to talk about it later, I'm a big fan of debris flows as long as they're not taking away my house. I've also done a fair bit of work looking at things like acid mine remediation. Right here, this is a stream that shows severe effects of acidification. This one happens to be in Kentucky. But because I'm working with the Forest Service, I've had a chance to look at a lot of the insults on the lands that were inherited by the Forest Service. You may have heard the term, the lands that nobody wanted. A lot of the Eastern National Forests are in fact lands that the Forest Service more or less inherited. They did take some, by various other means, but a lot of them were just simply, we've assumed ownership for one reason or another of these lands. And of course, the Gary Larson cartoon on climate change. I still think that we unfortunately function as if we all have the brain the size of a walnut, but I think we're working on that. There it is. Okay, one last shameless plug. I finally, after 10 years, my co-authors and I came up with this book, Reflections on Forest Management. Can Fish and Fiber Coexist? And I'll sort of cut to the chase. It's 500 pages, 13 chapters. I only wrote one, but I had hands in several others. The answer is, well, yes. And the answer is no. And the answer is it depends. So it's probably no surprise that fish and fiber do coexist. By fiber, I mean any tree that's cut for pulp or for lumber or for any reason whatsoever. Basically, tree harvest sort of writ large. And I have a chapter in here on the southeast. It features a number of the phenomena that are common to the southeast. So many books that had been prepared prior to this one focused on the Pacific Northwest. Washington, Oregon, Alaska, British Columbia, even the Rocky Mountains. And the South, oh yeah, the South is actually the breadbasket, but we don't want to talk about that. So in this volume, we talk very definitely about the South and the effects on the South. And such things as the keystone species of trees that are found in the South. It's not just the sequoias in California. It's things like the longleaf pine that used to be over huge swaths of the southern landscape, including parts of southeast Virginia, shortleaf pine across the Piedmont, and of course, bald cypress. All of these are keystone species that truly influence the habitat on which you find them. And then, of course, the historical aspects of that. And right here, one of the big reasons why the Fish Atlas for the state of Virginia is depauperate when you get up to the high elevations of the Southwest Mountains. It's because the streams were used as the conduits for transporting logs and other materials. That's arguably not good fish habitat, and that is from Southwest Virginia. So I don't get any royalties from this because it was written when I was on official time, but I do think that it is a nice compilation, a nationwide compilation talking about forest management and the conflict between fish and fiber. One of the other things that I've spent a lot of time on in my career is working with best management practices and how to ameliorate the effects of logging and land use in particular on fish and aquatic species. Okay. There, okay. Now, eels. How did I wind up getting involved with eels? Well, I mentioned that I've done a fair bit of work with best management practices and with effects of forest management and being responsive to the needs of the National Forest System. And back in 1998, I think it was, the forest biologist Don Kirk with National Forests in Virginia said, we have been approached by the Atlantic States Marine Fish Commission, and we need to know where anadromous fish are, in particular American eels are, on the forest. And I'm thinking, American eels? You know, I've been here 10 years. I haven't seen a one. Okay, well, we'll do this. We'll take your money, basically. And we sampled 10 streams along the Blue Ridge. And the team came back after sampling and said, oh my gosh, you won't believe it, how many eels we found. And in particular, in this one drainage, the Tye River drainage, which is right down here, just the other side of Raffine, just a little ways down the road, we caught in 100 meters, we caught, like 60 eels. It was like, holy cow, who knew? So this really started the wheels turning, and what are they doing? And this is over 2,500 feet elevation. It's getting up there when you're on the east side of the Blue Ridge. And we thought, well, we need to do a study on this, a more comprehensive study. So through Virginia Tech, we got a graduate student involved, and we started to do some research. And I'll get into that in just a second, I think. Yes. But I want to set the stage. It got me thinking, and then I realized that, I'm not the first person to be fascinated by eels. Rachel Carson in 1941. This is from one of her first published works in the Atlantic Monthly. It's a letter that she wrote to the editor talking about why did she use the American eel, or the eel, as an example of a critter in this book to characterize several of the oceanic environments that she was interested in. And it's because of this, I think, that there are strange places that an eel has been, places which I, being merely human, can never visit. And she was referencing the deep Sargasso Sea down several hundred meters. We didn't know how deep at the time, 1940, but we knew it was out there somewhere, and it had to be deep because we hadn't really seen any. Just as an aside, I was influenced greatly by Rachel Carson. I met her. I can't say I knew her because I was only like 6 years old. But my dad was a caretaker of a cottage that she owned on Southport Island, which is adjacent to Boothbay. And he would open and close because Maine cottages, they freeze in the winter. Have to go drain the pipes in the fall and make sure that they can take water in the spring. And when he went over to open the cottage, I went over with him, and Miss Carson was there, and I got to meet her. Didn't understand the significance of that for another 20 years or so. But it really, thinking back on it now, was like, wow, she was just a normal person. It's almost hard to believe. She wasn't a normal person, I should say, but to a 6-year-old, Okay. So to begin the story, it's important to understand, I think, the life cycle of typical fish that make long migratory journeys. And the easiest one to comprehend, and the one that I was most familiar with, was that of Pacific salmon, and salmon in general. Salmon are anadromous, meaning anadromous, they run against, so that the salmon spawn in fresh water and then go to the ocean, the eggs, they hatch as fry, they'll stay as par, and then all this other stuff is a little bit of noise. It's just the variations that you see on the theme. If you're a coho salmon, the fish I spend a lot of time talking about, learning about, and you're in Alaska, you might spend 3 years before you get to this stage, the smolt, and there's a whole lot of changes that go on in the body of this fish. And then you may spend an additional 3, 4, or 5 years before you come back to the same stream that you were spawned in, and you spawn and you die. And the carcasses, there's a whole literature on this. The carcasses of the salmon are actually responsible for some of the great productivity that you see in the streamside areas of places like Oregon, Washington, British Columbia, Alaska, Northern California. Virtually any place you've got anadromous fish, and you've got fish dying and being taken up in there. Bears eat salmon, and even though they may eat them, they poop in the riparian zone. So it's really enriching. It's a very rich environment out there. And it's complicated, but relatively well-known and straightforward. We know, for example, where in the ocean most of these stocks of salmon go. We know the timing of them. Not every one, but in general, we know how the system works. There's a lot of stuff that we have to keep track of and tweak. Things like El Nino and La Nina years. All of that sort of stuff can really complicate the picture. But basically, we know what's going on. Contrast this with something like the Anguilla species. And I put both on here, the American eel, Anguilla rostrata, and the European eel, Anguilla. Interestingly enough, even though the American eel basically are found they actually go all the way up here to southern Greenland and down, and all the way down to the Caribbean, and then down as far as Suriname. That's the American eel. The European eel goes over to parts of North Africa, Spain, the British Isles, all the way up Norway, Sweden, etc. What's really interesting here is all of them, American eels and European eels, go to the same place right out here, the Sargasso Sea. I don't know where exactly. The southern half is American eel territory. The northern half is European eel territory. How do we know? that's out of my scientific wheelhouse, but I have been told from reliable sources that they apparently don't mix, and that it takes, for example, the American eel, maybe as much as 1.5 to 2 years to get where they're going on the East Coast, maybe a little about the same to get down to here. It's where they spin off of, there's actually 4 currents that meet here, the Gulf Stream, one across here. I can't remember the names of all these. So anyway, 4 currents that keep this system in motion, and the eels peel off. And I don't know what cues they have, what it might be, but the European eels may take 3 years to reach their destination. As far as we know, they spawn somewhere down 400, 500, who knows how many meters, but a long ways down in the very, very depths of the ocean, of the Atlantic Ocean. The eggs are mostly buoyant, not super buoyant, but I'd call it semi-buoyant. They gradually make their way to the surface in amongst all of that sargassum. I've never been in the Sargasso Sea, but I've seen pictures and I've heard descriptions. It's really soupy, out there. And they're out there, and then they hatch, and they become leptocephalus, which is leaf-like. Sort of compare it to a small willow leaf. They're transparent, and they're out there in the sargassum until they start to peel off and make their way with the other plankton soup up the East Coast or down towards South America if they're American, and likewise to the east if they're European. Well, again, it may take them up to 2 years to get, let's say, up as far as Gaspé or something like that. As they approach the land, they start to metamorphose, and they go from this leaf-like leptocephalus to a glass eel. So that looks like an eel, only it's about yay big, maybe 2.5, 3 inches long, completely transparent. You can see all the vertebrae, you can see the guts, you can see the heart, all of that stuff. It's pretty wild. And as they approach fresh water and they get into the estuaries, they start to pick up pigment. They become somewhat more pigmented, more of a yellowish, greenish sort of a hue. Now, those that start to go into fresh water have to perform what I call a really amazing feat of magic. Their kidney goes from one of excreting excess salt and conserving fresh water to one that has to conserve salt and excrete water, because that's going from salt water to fresh water. And it happens really, really fast. Pretty cool stuff. Now, most of those glass eels that become elvers and they start to become yellow in color, a lot of them will stay right there in the estuarine areas. They tend to be mostly the males, hang out in the estuaries and not too far upstream, not exclusively, but mostly. The females, however, tend to start a journey upstream, and some of these journeys can be extremely extensive. One thing, and I'll mention this again later, is pretty well established that the further upstream you go, the larger you tend to get, the more likely you are to be female. And there's probably a real advantage associated with that. I'm not sure what. Maybe there's more exclusive food resources, although is that offset by the danger inherent in moving through these long stretches of unknown territory? Because I should mention this now, everybody eats eels. Everybody from blue whales as part of the plankton to juvenile fish to larval fish out in the ocean, in the estuary, other fish. And once they start to get a little bit bigger, more juvenile estuarine fish. Again, everybody, if they can get a hold of it, they'll eat an eel. And you probably realize that if you go to the coast, you can, or used to be able to go anyway, and buy eels, and they make wonderful bait for surf fishing and for even freshwater fishing. So they're running a gauntlet from the time they float on the way to the surface in the Sargasso, all the way through their life. They're running a gauntlet. It's also a good time to mention that the estimated minimum population size, viable population size for American eels is somewhere in the neighborhood of 700 million. That's the spawning population. And that's what experts, again, not me because I'm not a numbers person, say is necessary to sustain a population that might be distributed all the way from Suriname up to Greenland. That's a lot of eels. But that's actually down from what it probably was not that long ago. And again, when you consider the distance, because it's not shown here, and I actually show this, maybe I'll go ahead and show that. Yeah, I'll show the next slide. Or not. There, okay. This is the range in North America, and you can see that they go up the Mississippi. So there's eels in Arkansas, there's eels in Missouri, Nebraska, all the way up, up Wisconsin. They're all the way into there. Now they're greatly reduced from what they used to be, but that's an extremely widespread animal. Oh, I didn't mention there are 19 species worldwide of eels in the family Anguillidae, and actually in the genus Anguilla. Australia has 3 species. There's 2 in Asia, Japonica, and I forget the other one. But there's a bunch of different species. The American eel has become really an object of interest, and I'll talk about a little bit why a little bit later on here. But again, you can see that they're pretty widespread. So 700 million adults spawning. Let's just say that half of that is 350 million. That's the number of females. Each female, on average, will have several 100,000 to maybe 1 million eggs. So what's 350 million times 1 million? That's a lot of eggs. But again, think about how many a blue whale might take in at one fell swoop, so to speak. So it takes a lot of progeny to make sure that the eel can maintain itself throughout its range. Okay, so we get the female going upstream, and they may stay upstream. We find them, well, like in the Tai River, and I'll talk again about this, all the way up above 3,000 feet. And they may stay there for 15 years, 20 years, who knows exactly how long. In captivity, the longest lived eel has been over 100 years. And that's because that eel that was kept in captivity was prevented from completing its life cycle. Now, if you're in a stream and you can complete your life cycle, there's a queue, and nobody knows exactly what it is. You might be 5 years old, you might be 20 years old. And when that cue comes, it says it's time to move. Just prior to moving, the eye of the eel starts to get bigger, noticeably bigger. There's a layer of guanine that develops behind the eye. It's called a tapetum lucidum that actually helps to gather light. So that, this is an adaptation you would think for going someplace really dark. And that's ultimately where they're headed. They turn from a counter shading of yellow, black on the top and yellow undersided, to silvery on the underside and black on the top. And you can think that from above, if you're looking down on the eel from above, you see that black and you don't see the green. They're hard to see. They're cryptically colored. When they get to the ocean, if you're underneath, which is where most of the danger is going to come from, you look up and you see silver if you're a predator. And if you look down from it, you see black. So they completely change their body morph, their coloration, and when they get to the ocean, that kidney changes again. From now one that wants to conserve water and excrete salt so that they can make that long migration. And they make that migration whether they're from Canada, Suriname, or Virginia, all the way out to the Sargasso. How long does it take? Don't really know. I do know, though, that I had a graduate student try to track one, and we'll talk about that in a minute, too. It's almost mission impossible unless you've got an airplane and a lot more resources than what we had. Oops. Doggone it. There. So what are the threats to the American? Well, you can think, well, it's dams, obviously, particularly the high vertical face dams or hydro dams. And hydro dams, the biggest threat from hydro dams is eels going through turbines. Not a good thing. You wind up with sushi a lot of times. And there are a number of places in Massachusetts, Connecticut, New York, I'm sure in Virginia too, where they've been monitoring eels for a long time, and it's like, diced eel. It's just not a good thing. There's a whole industry developing now around how to design turbines such that the eel will get swept up in whatever the veins or whatever, and then spit out and not chopped up. So that's a growth industry because of the number of dams that are extant. Habitat degradation and loss, that's a big deal. Hydropower turbines, I just mentioned, and over-harvest. This is what a handful of glass eels looks like. And in the springtime, there's a relatively small window, and I can't remember exactly, it's April, May, maybe late March or something, that they start to move in, these by the hundreds of thousands. And this is, somebody mentioned at dinner, that the most highly valued per pound fishery in the world is for glass eels. You might get 500 to the pound of these things. They go for well over $2,000. There are only 2 places in the U.S. where you can legally harvest glass eels, Maine and South Carolina. Every place else, it is not legal. The incentive, however, is so great that you can go out with a dip net and get probably 10 grand worth of eels easily in a couple of hours. You then have to market them, and where do you market them? Most of the glass eels right now that are harvested in the U.S. go to either Japan or China. The bulk are going to China because they pay the greatest price for them. They use them in grow out for aquaculture. They can grow an eel to about 340 grams. It's about 3/4 of a pound, which is the size that's suitable for producing kabayashi, which is the Japanese-style barbecued eel. And there's a huge worldwide market for that. They also produce smoked eel or unagi for the sushi trade. 80% of all of the eel that's used in sushi or barbecued eel comes from China today. There are a few other places in the world, a little bit in South Korea. Japan has some. There's a small startup in Maine called American Unagi that's hopefully going to come online because they'll be able to take the main, I hesitate to say grown, the main captured eels and at least keep them in state, keep them in the country. And the other thing is they're regulated by the FDA. Eels coming from China are not regulated by the FDA. And there are some things anecdotally that I have been told, you won't find these in the literature, that are done in aquaculture in China that have kept me from ever wanting to eat unagi on sushi again, unless I know that that's not from China. And it's for 2 reasons. One, because who knows what they're feeding them. You've all heard about the melamine stuff. Eels eat almost anything. So it's not that big a deal. The other thing is to keep eels in captivity at densities great enough to harvest and make money on. It's problematic because in great densities, eels tend to become male. And males don't get big enough to fillet and barbecue. They just don't. So you want a preponderance of females. And there's 2 solutions. One, have them very, give them their own tank and everything will be fine, and they'll probably develop into females and get big. You can't do that and make money. The other solution, use estradiol, a feminizing hormone. And you can do this, and you will keep roughly 60 to 80% of all the eels, regardless of density, female. And economically, that's a huge advantage. Now, there are several forms of estradiol. None of them are approved for aquaculture in this country, at least not yet. That same company, American Unagi, has partnered with a university in Canada to develop a form that would be acceptable and to make sure that all of that is cleared before anything ever enters the market chain. It's not cleared yet, but if it does, then that market should take off. In the meantime, you're rolling the dice because that estradiol may have been derived from who knows what when it comes from some of these other places. And again, people say, oh, it's probably safe, but that's not safe enough for me. So you can take that for what it is. Maybe you still want to, it's sort of like eating tuna and getting mercury. If you eat it once a week and you're pregnant, probably not going to have a problem. Eat unagi once a month, probably not going to have a problem, but I don't want to encourage the market, so I'm not going to do it. Anyway, enough said about that. So let's talk a little bit more close to home here. This is work done by Nick Walker, George Mason University, and we sort of go through what are the more likely reasons for the decline seen in the landings of American eel. This is for adult eel now, for the adult market. And it's pretty clearly, one of the big actors, bad actors, are dams. 1800 to 1992, look at the increase in dams. From about, what is that? Just before I was born down here until, well, when I graduated from college, that was the heyday of dam construction in Maryland and Virginia. It just took off like a rocket. And just as an illustration, you can see where the dams are, most of them. And there's, I don't know if you can see the white docks. It doesn't really matter because there aren't a whole lot of them. These are the dams with fish passage. Most of those fish passage dams were put in for things like striped bass and alosids, the shads, the American shad and alewives, things like that. Eels will use them, certainly, and eels, but in many cases, eels don't even need them. They can go around a lot of these dams. But this is, if you want to point your finger at one thing, this is a pretty good place to point. Okay, so with all of the stuff that threatens the eels, should they be imperiled? Well, several times they've been petitioned to list. 2004, 2007, 3-year study period, listing was not warranted. They were petitioned again in 2010, and listing was not warranted. Now, I have to confess, I was one of the reviewers on this plan. And it wasn't so much a plan, it was a compilation of the data that could be used to justify a listing. And the problem is the issue of American eel declines is very much watershed by watershed. Now that's big watershed, like the Hudson versus the Shenandoah versus the Potomac, that kind of thing. But nonetheless, you can have adjacent watersheds where eels are doing fine and other ones where they're not. So does that warrant being threatened? The other thing is, because eels are right now considered panmictic, which means that genetically you can take an eel from South America and one from Canada, and if you do a genetic profile, you can't tell the difference. It's not like salmon. You take a salmon from Southeast Alaska, one from two different watersheds in Southeast Alaska. You can tell them apart, but you can't do that with eels, at least not with the techniques that we have now. So how do you justify, on the basis of genetics and population purity, calling this thing threatened when it's doing just fine here, even though it may not be doing so fine over here? Well, the thought is that maybe we'll get more refined in our genetic analysis, but as of right now, it has not been listed. In Canada, it's threatened. Threatened doesn't mean not harvested, because there is a considerable eel harvest in Canada. It's tightly regulated, but there still is a harvest. The IUCN Red List, that's worldwide red list, they list the American eel as endangered. And I think that's, well, I'm pretty sure that's partly because there's a large but unknown traffic in glass eels. So we know legally, well, mostly legally, what's coming from Maine and South Carolina, but there are illegals, eels harvested illegally all up and down the coast. And in the Caribbean, there are people who are openly going to the harbors and buying eels. So it's a big deal. And it's because that fishery in aquaculture is so valuable that they're willing to pay these high prices. And data from the Caribbean is scarce. We'll talk a little bit about that. But how did I get involved? Well, I mentioned earlier that it was because of that study that we did for Don Kirk on the national forests in Virginia. So from 1999 to the present, we've been interested in American eel ecology in headwater forested watersheds of the George Washington Jefferson National Forest. 2011, we started doing work in Puerto Rico on eel distribution and abundance in 5 watersheds of El Yunque National Forest in Puerto Rico. I'll show a map later, but this El Yunque it's not pristine, but it's relatively intact rainforest. And if you look at an aerial photo of Puerto Rico, the El Yunque stands out, wow, like a thumbprint. But anyway, they also have American eels down there, and we're interested in finding out about those critters. The work on prioritization of watersheds, that was done by Nick Walker at JMU. American eel distribution above and below dams, because Puerto Rico decided we need to do something about eels. What are the threats? Well, let's look at dams, fish above and below. Shortly thereafter, Hurricane Maria and Irma hit, and that made us going down there for a whole different reason. And then 2018, we started work with eDNA as a tool for large-scale detection of American eel presence and absence. So that's a bit of a summary of the kinds of stuff that we're doing. So for the Virginia work, the background and objectives, headwater stream survey, as we said, eels found in several streams in the summer. We went back in the fall of that first year where we'd found so many eels and said, well, you got to show me. So I went out with the crew and we electrofished. We didn't find eel one, right where they had found 60 before. What is going on? So we thought, well, perhaps those eels are moving amongst habitats. Maybe this was summer habitat. Maybe there's a winter habitat. We don't know. We really didn't know anything about it. So in 2000, we started a graduate project. Andy Strickland, a graduate student of mine, looked at habitat preference, density, growth, and movement of American eels. In this case, it's in Tye River and a couple of the tributaries. We've carried on that study since then with annual sampling for long-term monitoring of growth, and we'll talk about that in a sec. Come on. This is just a schematic. This is where the Tye River and Shoe Creek are, James River here. I put this on here because serendipitously, we've been doing a trout stream study up in Stanton River, which is in the upper reaches of the Rappahannock, and downstream of that, there was a dam removed, the Embry Dam at Richmond, and since removal of that dam, I've got a figure to show you that deals with what happened within a very short order of removal of that dam. There, the methods. Eels are elusive, so instead of doing the typical 100-meter reach, we did 2 kilometers, 2,000 meters instead of 100 meters, because we wanted to be able to, if fish were going to move within a certain distance, we wanted to be able to detect it. Periodically, we did a kilometer below that and a kilometer above that, so we had a total of 4 kilometers that we continuously electrofish. So if they were there, we were gonna find them. We did multiple pass electrofishing, as indicated by this here. We have an annual eel rodeo. And by the way, if anybody wants to volunteer, in July, generally the week after 4th of July, we have our annual eel rodeo down there. It's right just below Crabtree Falls. And we get anywhere from 20 to 30 people. And we put everybody to work because they're not that easy to catch. They're not that easy to see. And they're not that easy to process. I mean, they're problem childs, but they're really fascinating. So we use a combination. Early on, we used radio telemetry. And then more recently, we're using these passive integrated transponders, these things, PIT tags. These are the tags that people put in horses and dogs and cats. There's a 10-digit alphanumeric tag that identifies that particular fish or horse or dog or whatever it is. We inject it into the body cavity using this thing, a really sharp needle, put it in there, and it stays there. It's inert, and then we have a detector, and we can wave the wand over it, and beep, if it's tagged, we can tell, this is eel, AA, blah, blah, whatever it is. And these work really, really well. They have revolutionized a lot of the mark recapture studies that we're able to do, not only on eels, but in a variety of other species as well. So there's a video here. Can somebody up there in the control booth? It's okay, I'll narrate. This basically is, we've done about 20 meters of sampling, and the water's a little cloudy here because we've clouded it up, but that's a typical catch, and that's what it looks like. We anesthetize these things with, it's called eugenol, but you can go to the health food store and buy clove oil. It's the same thing. And that anesthetizes the fish, plus your hands smell really good when you're working on that. And they recover really, really fast. It's not approved for use. It's sort of ironic. The approved drug is what's called tricane methane sulfonate. It's a barbiturate, right? That's approved. And, you know, it's like, I don't want to put my, anyway. So we're using this one. We're using this under a special use authorization. I am a master permitteer, which means that I get to use eugenol. And a lot of other folks around want to use it, so I can sub-permit them. They have to keep track of how many fish, where, what temperature, all that sort of stuff to build up a database. And ultimately, this will be approved because, you know, you can almost drink this stuff. And it's such an effective anesthetic, and it has such minimal effect on the critters. That's a bit of an aside, but kind of interesting, I think. Oops. Okay, they get it. Let's go. There, okay. So Strickland's result, the master's student. Habitat preference with telemetry, not a surprise. Deep pools and large substrate, they really like this. Greatest movement was greatest in the summer, least in the winter. Daily activity is greatest at night. You know, they really, if you go out in a typical trout stream, you don't generally see eels unless there's something odd going on. But if you go out there at night, you might see them because that's when they're hunting. Mid-fall to mid-spring, eels move very little. They shelter in the substrate and under stream banks. This answered the question of, well, where did those eels go? So we would be sampling. We had a bunch, I think we had 15 eels with radios in them, and we followed them, and they went just to large boulder places in the stream. And we at first were thinking that they were dead because we go out there, you know, once every hour for 24 hours, and they didn't move. Well, it's got to be dead, right? So this is in December. I did the right thing. I had my graduate student put a dry suit on and get down in the stream and start digging because we thought we're going to find a dead eel and get our radio back. He goes back in at arm's length, pulls a rock out, and he's looking, and there's an eel looking at him. And that eel shot out, and he just right at him. It's like, whoa, came up out of the water. It was pretty cool, though, because I had the antenna on the bank, and if that's the stream bank, I'm standing back here, and it says the eel is underneath my feet. How can that be? Well, let's look. And sure enough, that's exactly where it was underneath my feet. Now, this has management implications, right? Because streamside management zones, we like to have a permeable, what's called hyporheic zone, an area where there can be water exchange and critters actually live in here. So we can't allow sedimentation not only in the stream, but also in the riparian zone. We don't want that because it packs those spaces and it does away with habitat, clearly for something like American eels, but there are a whole host of other critters that would live back in there too. So there's a good reason for not building roads right up next to the stream because of that compaction that can go on. Not a good thing. So that was an unexpected bonus to this particular piece of the study. And we got out of this density estimates, population estimates, a thesis, all of the good things, plus the beginnings of a long-term study. And again, this is the big thing. Where do they shelter? Mid-fall, substrate, and under the stream banks. Really important stuff. They didn't go anywhere, not really. They went a few meters, just they found a good place to hang out for most of the winter. What's interesting about this is during the winter, you know, again, I was a real hard something or other major professor, get out there and every hour in the winter. So started at like 6 a.m., nothing. Gets to me midnight, he gets out there and then at 1 a.m., boom, all of a sudden the eels come out, they're foraging, who knows what they're doing, they're going all over the place, and then by 4 a.m., back under the bank. So there's like a 3-hour window of activity in the dead of the night or the early of the morning, and then right back in where they were. So they don't do the black bear thing. They don't go in there without any food. They're probably eating. They're getting something, but it's on their schedule, and it's not something that's readily apparent to us mere humans. Okay, this is another little bit of a video. Well, this is me narrating it. This says American eel, 2.5 miles up the Stanton River. And I'm saying that that little critter, it's probably 4 years old, meaning from the time of an egg until now. It floated around in the Sargasso, got to the lower James, headed up the James, and now 4 years later, it's finally exposed itself to capture by us rookie fish biologists. That one is not quite big enough to put a pit tag in. They've got to be at least 200 millimeters to put a pit tag in because they're so thin, about as big around as your little finger. If you put a pit tag in there, it just won't fit. So we've got a limit, you know. So every year we're really interested to see all of these because it says that there's some new recruits coming in, but we can't always tag them. Okay, for size, I'm going to get a little wonky now. Not too wonky, but a little bit wonky. This is based on, you know, just under 4,000 eels, the average length for Virginia streams, and I'm going to call, in this case, Tye River, Virginia stream, 376 millimeters. Shoe Creek, we had a few less. Shoe Creek was a victim of budgetary constraints. We just couldn't keep going back in there. It got to be too expensive. And then if you look at weight, the average weight of these things, 86 grams, that's not a huge eel. But it's still really, really interesting. And it's very consistent with the comparison data from some other studies. I won't dwell on that. Now, this is what's interesting, the range of the eels. And by the range, I mean when we captured them to when we recaptured them. Where were they when we captured them? Where were they when we recaptured them? Because they were marked with a pit tag, we could use GPS and say it's at this particular spot, and we put that into the database. We catch it again, we query the eel, we read its tag. Well, last time it was here. And how far is it from where we tagged it? In many cases, it's within, 100 meters of where we tagged it. That could be a year later, could be 2 years later. And if you look at the radio telemetry work, it's very consistent with what we were able to track on an hour-by-hour basis that Strickland stuff did. So it's pretty interesting. Once they get marked, once they set up a housekeeping in the stream, they tend to stay there. So the only thing I want you to look at on this pretty complicated looking graph, it's not really complicated, is this. This is the year of tagging. So the year 2000 is when we started, when we had enough eels to really do tagging. Over the course of 22 years, that's since last July, we have recaptured 385 eels. That's obviously more than we tagged. So we're getting the same eel over and over again. From the standpoint of fish biology, this is really fascinating because we fish biologists like to compute things like mortality on the basis of mark and recapture. So that if we mark 100 fish and we catch back 20 at a later time, then we think, oh, there's probably been some mortality, maybe 80% mortality, something like that. We would make some estimates about mortality. It's not quite that simple, but it's something like that. However, if you think about the number of fish that were likely alive, at the time of your first recapture attempt, in other words, you tried to catch this eel, it wasn't there, or you didn't catch it. One of those is true. Look right here. There's an eel that was tagged in 2000. We recaptured this many eels for the first time of that batch. We thought, wow, 12 years later, we had not seen that eel since the year 2000. That's cool. And it was really, really close to where we had tagged it. But that was nothing. In 2017, we got this eel. That eel was within 30 meters of where we had tagged it 17 years before. Just amazing site fidelity. And again, it found its place, and that's pretty cool. And again, you can see that if you look at the overall percent of recaptures, first year recaptures, we're not causing mortality, and the eels themselves don't appear to be dying, and they don't appear to be moving at a particularly great rate. If you want to take a deeper dive, I love going in there and pulling out numbers, but those are the big ones for purposes of this talk. The percent of recaptures, the more years we sample, the higher the recapture rate. Long-term data, wow, there's nothing like it. You know, here we are in 2022, we're still getting the percent recaptures going up. This is the overall percent of recaptures going up. And you know, if you had a 3-year study, you're in and out, you might think, oh wow, look at that. You'd have something more along the lines of this. It takes those long-term data to really get a sense of the range of samples. Tagging small eels. In other words, if you tag eels that are on the smaller end of the spectrum, that can pay real dividends. If you tag a 400 millimeter eel, the chances that it's going to become an adult and go out to the ocean are greater. So eels recaptured 10 plus years later when they're less than 400 millimeters at the size of tagging. So tagging the little ones, if you want long-term data, that's where the biggest dividends are. And here's one that I particularly like. This is that Stanton River that I showed, that's the Rappahannock tributary. Here is sampling. We were capturing American eels right up until the time that the dam was removed down at Richmond. Come on. There, okay. Dam removal. Well, it took a year or two, but then you can see, you know, once the momentum built, this is eels that were captured incidental in a trout stream sampling project. That's the number of eels per year in about 6 kilometers of the Stanton River, from roughly where the Stanton River confluences with the Rapidan River, just inside Shenandoah Park. A huge increase in the number of eels and the number of larger eels too, because those eels are doing just what they do in the tie. The longer they stay there, the bigger they get, and the more frequently we see them. And I think this is another video. Yeah, that was from last fall. That's the largest eel we've seen in the Stanton River ever in 29 years of sampling. So it just points up the difference. You know, we now have viable eel habitat up on the east side of Shenandoah Park. And that one weighs 380 grams. I just happen to know that, which is about the typical size that you use in aquaculture that could wind up as barbecued eel somewhere. This guy didn't. We put a tag in him. Actually, we didn't put a tag in him. We let him go. We don't have permission to tag the eels in Shenandoah. Variety of reasons. There. Conclusions. Eels are highly mobile or highly sedentary, depending on the life stage. They don't move much in the headwaters. They stay a long time. Local conditions are important. Long-term studies. And small eels return to Stanton. Highly mobile. I say this because we did have one eel that, with the radiotelemetry study, everybody else was staying put. This one all of a sudden had moved 200 meters, and it was downstream. Oh, what's going on here? The next hour, it was 1 kilometer downstream. And it was like, whoa, it got its walking shoes on. So Andy, the graduate student, started following this critter, and he followed all that day and into the night, and it was down about 10 kilometers, and he had to stop, and the next day he couldn't catch up with it. It was going that far, that fast, on its way down, presumably to the ocean and on out to the Sargasso. So when they get a notion to move, they're going. It would have been nice if we'd have had an airplane, because we did have radios that could have been enabled if we could have flown, but that was beyond our budget. Nonetheless, pretty cool stuff. So American eels in Puerto Rico. While all of this is going on, we started sampling in Puerto Rico. It was prior to 2005. They're known to be present, but assumed to be low density and only found in a few streams. Tom Kwok, who was late of NC State and the U.S. Geological Survey, had done some work down there. He found eels in many of the drainages. Most high densities are in the northeast. This is of El Yunque National Forest. Yeah, and here's Puerto Rico. Here's El Yunque, And this is what El Yunque looks like from above. Pretty cool habitat, and we went from the ocean on up, from ocean to mid-level to upper level, and all of this occurs within about 15 kilometers. So it's a really nice, compact system to be studying American eels. Goals: find the upstream extent, estimate population density, compare with the Kwok results. Basically, I'll cut to the chase here. Dams and falls make a big difference. Dams, there are still eels found above dams like this, and above this fall, we did 3 passes. We didn't think anything. The top of the fall is actually there. It's pretty high. We didn't think we'd find any eels above that. We sampled above that. 1 pass, nothing. 2 pass, nothing. 3rd pass, there's an eel. So they were able to make it up and around. As I said before, they're able to navigate around places like this when it's really wet, find purchase and get around falls and sometimes even dams to get upstream habitat access. Conclusions of that study: it takes a watershed to raise an eel. Barriers are key. Public education is necessary. The Puerto Rican public doesn't really realize, except for those that are harvesting the glass eels. How long do they stay in the streams? New study questions. What is the eel growth rate? Another question. Right down at the bottom, the impacts from hurricanes. This was a question that arose. Well, it turns out we were able to answer that right after Hurricanes Irma and Maria. We had just done a whole bunch of work. I've been lucky in my research career. I've gotten into places right before catastrophic events like hurricanes and debris flows. So I have before and after data. You can see challenges to infrastructure right here. Defoliation, massive, large wood loads. Tabanuco, that's a champion tree that was defoliated. What's interesting, one of many things that are interesting, landslides, 250. This is on the forest, by the way, not Puerto Rico-wide, just on the National Forest, 250 landslides in roads, 3 times that in streams. You can see that habitat has been completely taken down to bedrock in many places here. But there is resiliency. I was down there less than 6 weeks after Maria, and already the Colorado palms were leafing out again. Their strategy is when the winds come along, you drop your leaves, because if you have leaves up there, you're vulnerable to wind throw. Likewise, you can see how green this is within 6 weeks of that hurricane. That's a very resilient place. It makes sense because hurricanes are not a new phenomenon in the Caribbean. Fish-wise, little fish we found were returned to the areas they had been. Big fish, these are all Puerto Rican species. This is the mountain mullet. All sizes were present. Some of these shrimp, these critters, this is a macrobrachium shrimp, and yes, it is harvested, and yes, it is good to eat. It's a problem for it. Amazing colors on some of these. Lots of sizes of shrimp. This is a shrimp. It's a crustacean-dominated system, so there are more shrimp and crab species than there are fish species. But the American eels were there, as they had been before, all sizes, including that one. That's about like a 4-pound American eel. So they're really, they're pretty impressive in these streams. One of the bad things we found, all of these are exotic invasives. The cichlids are from Africa, tilapias from Mozambique, Central America, the jaguar, Amazon, obviously South American catfish, red devil also from Africa. Probably aquarium escapes, but we think what happened is during the hurricane, they washed down to the estuary. They went up watersheds where they hadn't been before, so they found an avenue for invasion. So that was not good news, but it's good to know right at the beginning. Graduate student at NC State, Ambar Torres Molinari. She just finished her master's this last fall in Puerto Rico, following up on some of the long-term eel work. And again, you can see some of those eels are pretty substantial size. I'll cut to the chase on some of these. American eel total body length. Again, you can see the greater the body length, with one exception, the further they are from the mouth of the river. Total body length and the probability of being female. I mean, it's pretty clear once you get to be above about 320 millimeters, you're almost certainly going to be female, and you're going to wind up being higher up in the watershed. in Puerto Rico, just like every place else. The density of fish goes down. It's still pretty high, but it goes down the further you go from the river mouth, not surprisingly. And one more. Oh, that was it? I thought there was one more. Well, it's in terms of the adult population, it's pretty good news for Puerto Rico. The biggest challenge they face going forward is that fishery, right now, illegal fishery in the glass eels or elvers. So, in summary, there's commonality in eels, whether you're in Puerto Rico or in Canada, and Virginia has served as a really interesting laboratory for us to explore the freshwater habits and life cycle and behavioral quirks, if you will, of being an eel in trout streams in Virginia. Tye River, by the way, is a great trout stream too. So the eels may eat the occasional trout, but you don't ever see a distended stomach of an eel that suggests that it's eaten a trout, certainly of any size. So with that, I'd like to thank you for your forbearance and welcome any questions.
Thank you very much, Andy. And we would like to take some questions. Let's make sure we use the microphones just so the folks online can hear the questions as well. And Joe has one in the back there.
Yeah. So how long do you think these eels that are in our streams here hang out before they go back to the ocean?
First of all, I don't know. But there's no reason to assume that it couldn't be, a minimum of 5 to 7 and up to 20 years. We caught an eel in 2022. We'd caught it before. That had been marked in 2000. So we know it's 22 years in fresh water. Add 2 years at a minimum. So it's 24 years old now, or was in July. And again, there's no reason why it wouldn't be here also. I think, did I say the record? Actually, the record was from it was from a single eel that was put into a well, pretty cruel, I think, in Sweden. And it lived over 150 years. I just can't imagine living in a well. Anyway.
Another question down here in the front.
Thanks. I was wondering what triggers the eels to actually go to the ocean.
Me too.
and when they go to the ocean, that adult form, I guess the silver eel, will they die after they lay the eggs, or they come back?
They either die or become sea monsters. we don't know is the short answer to that. I mentioned at dinner, it's several hundred meters down and 700 million eels. Wow, it must just be, I can't even imagine what that must look like. Fortunately, there's no light, so nobody has to see. But it must be something to see. Yeah.
It's pointing to you. Okay. Quick question. So if I'm understanding correctly, the eels, when they're in our mountain streams, are coexisting with our brook trout, and they are basically participating in this cold water
Brook trout,
fisheries
black-nosed dace, you name it, who's ever there.
So are the same threats to brook trout and to those watersheds threats to the eels as well? Or do they have an alternative? In other words, is this essential for their cycle?
The far upstream, I would say
Yeah.
the far upstream. I think the biggest threat to eels is access. And I would say that for any population, well, it used to be access for brook trout, but we have so few populations that are connected stream to stream. I mean, if you think about the Stanton River, it's connected to the Rapidan right at the confluence. And much below that, and there's not a whole lot of connectivity. But that's, if you go to a place like Payne Run, that's an isolated population. Payne Run basically goes subterranean before it hits the Shenandoah, the South Fork Shenandoah. And so many of the Shenandoah streams are, they are brook trout streams, they are isolated. And I think one of the reasons that, for example, the brook trout stream of the Stanton River, that it was not extirpated from brook trout is, first of all, the debris flow that hit the Stanton River was only like 3 kilometers up, so everything below was wiped out. But the colonization of brook trout and everything else for that matter came both from above and below. And we were able to document, we could see the different groups of fish coming to meet in the middle. Throughout this all, eels were probably there too. If anything can escape the effects of a catastrophic event, it's going to be an eel. So I hesitate to call eels a canary in a coal mine because they can take more than almost anything else, any other fish. They can be out of water for hours, like in the bottom of a boat, and you put them in water and they start swimming. They're pretty tough. They can take temperatures up into the 30s centigrade, which is, up around 80, 90 degrees. Not for huge amounts of time, because you simply do run out of oxygen when you get to water that warm, but they can take it. You put brook trout in 80-degree water, it's toast.
We have another question back here.
So, did you say that they keep getting larger and larger while they're in our streams here?
Based on
Each
what
year?
we've got, based on our pit tags, because
and-
when we mark it, we weigh it, we measure it for length and we weigh it.
Yeah.
We recapture it, we remeasure and reweigh, and we've never seen any either not grow or get smaller. That's not the same with brook trout. We've done similar kinds of work with brook trout, put in pit tags, and we've got some brook trout that we tagged 4 years ago, let's say, which means that they were probably a 6-year-old brook trout because they didn't get big enough to tag until they were 1 plus almost 2. And we have seen over time that fish might stay the same length or actually decrease slightly year to year, the brook trout. Not true of the eel.
So, could you tell us, instead of millimeters, inches and pounds, how a big one would be? How big a big one would be?
Eel?
Yeah, eel.
Let's see.
Before they go back to the ocean.
Oh, let's see. So a meter is roughly 3 feet. So anywhere from 24 to 48 inches. Boom to boom. Typical is probably in the neighborhood of 400 millimeters, which is, help me, Steve, is that like 20 inches or so? Yeah. And they tend to get fatter too, of course, which is, there's a premium on fat.
For those who haven't been to Riverfest in Waynesboro, we often do get them in shocking and get to see them and hold them. So they're right here in town.
Should say try to hold them.
Yeah, try to hold them, yeah.
They'll
Well, I
fill
think
a
you'll
bucket.
agree. Yeah, I think you'll agree. It's a fascinating topic. It's certainly something we want to interpret when this new museum comes. It's an interesting story to tell, and it's very local, also very worldwide. So appreciate, again, Andy coming and giving this presentation. I'm sure he'll be happy to stay around and take any questions one-on-one, and I hope you'll join us next month. Thank you.